I think it's very important to understand what it is that causes death, because we begin to recognize, as we understand these processes, that what they are perfectly natural biological and chemical responses to our surroundings. What happens in a heart attack is that the heart stops pumping. Either it stops completely, which is called cardiac arrest, or it starts a very complicated rhythm that is ineffective in pumping blood out. So really in a heart attack, what we have is the ultimate deprivation of the cells of their oxygen. And the cells die, and most particularly the brain cells die, because the pump is no longer effectively driving the blood to them.
It's useful stuff, this oxygen. But 2,000 million years ago, when it was all starting to happen, terrestrially speaking, I mean, neither of us would have stood a chance. You see, there was no oxygen in the atmosphere then, just nasty gases like methane and ammonia. And it wasn't until the first plant life showed up and started to photosynthesize that the oxygen began to appear.
It was perhaps the worst case of air pollution ever recorded on this planet, because the organisms living at the time were anaerobes. They thrived in a reducing environment, and then all of a sudden you presented them with an oxidizing environment, and they didn't like it. Present-day anaerobes are killed by oxygen. So presumably those anaerobes didn't like it either, and they either had to retreat to environments that the oxygen didn't penetrate, or dispose of the oxygen in some way, or they could maybe evolve to use the oxygen, but they also had to evolve protective mechanisms, what we call antioxidant defenses. Some organisms evolved surface enzymes that just used oxygen and reduced it to things like water or hydrogen peroxide, so it didn't get into the cell. But the big advantage was really the evolution of aerobic respiration, which firstly helped to use up oxygen, but also enabled organisms to very efficiently produce energy. Aerobic respiration is about 14, 15 times more efficient than anaerobic respiration.
If oxygen is now the very basis of life, it's still something of a double-edged sword. Babies born prematurely pose a special dilemma for doctors and nurses. Because their lungs and respiratory systems are underdeveloped, they need to be given higher doses of oxygen to sustain their fragile grasp on life.
Obviously, we all need oxygen to survive, but in fact, oxygen is toxic. And oxygen is toxic not because of its own reactivity, which is rather feeble, but because it's capable of being involved in a variety of reactions in the body, some of which occur normally, which can lead then to toxic species, as we call them. And these species are free radicals.
Over the last 20 years, there have been tremendous advances made in the ability to keep premature babies alive. So much so that many babies that are born around 22, 23 weeks gestation now are surviving. The problem we have is that we have to give these children an awful lot of oxygen. And we know that oxygen in high quantities will lead to increased radical production.
The body is made up of tissues and cells, and they're all made up of molecules. And within those molecules are electrons. And the important thing is that electrons like to be paired. They go around in pairs, and that's their stable configuration. A free radical is a molecule with an unpaired electron. So clearly, its task in life will be to find another electron to pair with to make it stable. So it will attack whatever's around, pull another electron from there to pair its own electron. But in so doing, will leave behind an unpaired electron. So this will initiate a cascade of reactions where electrons are being passed from one to the other, and in the process can cause damage to the cells of the body.
One of the things that free radicals do is it actually prevents those normal processes of protein synthesis and DNA synthesis. So the tissues don't grow normally.
A further complication is that babies born prematurely haven't had the time to develop their antioxidant defenses, the way the body naturally copes with the toxic effects of oxygen.
The body is brilliantly designed. It's constantly being attacked, not only by free radicals, but by all sorts of poisons that we eat in our food, in all sorts of poisons that we breathe in the air. So the body has to survive by developing a whole series of defense mechanisms against natural hazards. Perhaps the most beautiful one of these is the one that really deals with these toxic byproducts of oxygen. This is called the antioxidant defense system of the body. It comprises things like vitamin E, vitamin C, and a series of enzymes and proteins which are strategically located in different parts of the cell so that they can neutralize radicals as and when they're produced.
You can stop holding your breath, folks, to welcome the one and only oxygen! The life giver and the life taker away. This contradictory little character's the commonest element on the planet. Yet, sorry, oxygen, didn't mean to offend. A gregarious little creature, oxygen likes to link up with just about any other element you care to mention to form oxides. Oxygen goes round as a pair, but sometimes it turns trio and gains an atom, becoming another reactive gas called ozone, which at ground level is a pollutant. But high in the upper atmosphere, it blocks out the sun's ultraviolet rays and helps to prevent cancers. Yes, folks, there are two sides to every story.
Not everyone worries about their health, but I know a lot of you do. So you may like to know that two of the main sources of free radicals in our environment are sunlight and tobacco smoke.
They're such difficult things to study. They're only around for a few millionths of a second. You need extremely complex technology for measuring them, identifying them, looking for the fingerprints of their activity. And it really is new technology. I mean, in the heart field, it's really only been studied for the last 10 or 15 years. So one of the ways in which we study the effect of free radicals in the heart are to take single heart cells, isolated from a whole piece of tissue, perhaps from a sample in the operating room, study it under the microscope, and monitor many of the activities that you would see in a normal heart. Included in the fluid which is going round the cell, providing the oxygen and the glucose, there are photoactive agents, which when illuminated with a particular color of light, will generate free radicals. What then happens is these radicals, although they're only there maybe for a millionth of a second, will start attacking the membranous coating on the outside of the cell. They will start to oxidize fats in the membranes. They will start modifying proteins. The cell may start twitching, it may start contracting bizarrely, and eventually, if the damage is enormous, the cell will begin to round up and go from this beautiful rod shape down to a round, amorphous kind of ball. And that's exactly what happens during a heart attack.
One major killer in heart disease is what is known as reperfusion injury, when a heart that's stopped starts pumping again minutes later, bringing a rush of oxygenated blood to the starved heart tissues, and a burst of radicals along with it.
What you're seeing here is a piece of isolated heart tissue beating away, completely removed from the body. Now, in these experiments, what we will do is we will suddenly induce a burst of free radicals, and you will see a transition of a perfectly normal and regular slow heart rhythm into a very, very rapid one, which will then deteriorate into a state of complete chaos. That's called ventricular fibrillation, and that kills you. We know that those radicals are only present for a few millionths of a second, but in that time they can do sufficient damage to cause perhaps a lethal heart attack, a disturbance of cardiac rhythm, which would kill a patient in 2 or 3 minutes. We have other effects which take longer to manifest themselves, possibly in diseases like cystic fibrosis, where the lack of antioxidant defenses slowly builds up a cumulative injury in the tissue, which takes its toll with time. And then at the extreme end of the spectrum, we all grow old. And much of the aging process, the withering of skin, the changing of color, the changing of the texture of connective tissue, that is all a sort of cumulative effect of radical attack over perhaps a century.
Some of the most healthy foods. Halibut, first on the menu. Its oils are rich in vitamin D, good for your bones. Liver, lots of goodness here, but you don't need to eat it often. A single serving gives you a month's supply of vitamin A. Curry powder. You need iron? Forget spinach. Take a takeaway curry. Cheese. Calcium is vital for your bones and muscles. All you need is a daily serving of hard cheese. Fruit. You can enjoy a selection, but 100 years ago, sailors had to suck on limes to get their vitamin C. Brazil nuts. You need just a dash of selenium for the body's defense. 1.5 Brazil nuts gives you a daily dose. And that's 6 really healthy foods.
Cyanide, once it gets into the cells through the circulation, has within it the ability to prevent the cells from using oxygen in their chemical reactions. The trouble with carbon monoxide is that hemoglobin likes carbon monoxide a lot better than it likes oxygen by a factor of about 300 times. So if we inhale carbon monoxide, our hemoglobin will just suck up that carbon monoxide. It replaces all the oxygen. And a compound called carboxyhemoglobin is created instead of oxyhemoglobin. That goes to the cells. They're not getting oxygen, and they quickly die.
Georgy Markov was known to be a dissident. He was watched, by our people. Initially, the reaction was, by most people, that this was paranoia from a defector. This is common, people imagine they see spies behind every fence.
On Thursday, the 7th of September, 1978, Georgy Markov, a Bulgarian dissident working for the BBC World Service, was taking his usual route to work.
He had parked his car, and he was standing on the bus stop on the south side of Waterloo Bridge. And as he was standing at the bus stop, he felt a blow on the back of his right thigh. And on feeling this blow, he turned round to see a guy bending down, picking up an umbrella, which he'd dropped. The man apologised to him in what Georgy Markov said was an East European accent. The man then hailed a cab and disappeared.
Next day, he was admitted to St James's Balham, where he was noted to have a temperature, to have a high pulse rate. They saw this thing on the back of his leg, which looked like a small hole with an area around it, which was quite red in colour. And they also noticed that in his groin on the same side, there was swelling and tenderness of the lymph glands.
Markov died 2 days later, and the pinprick in his thigh proved to be the entry wound for a tiny metal pellet, presumably fired by a gas gun hidden in the dropped umbrella.
The pellet was 1.53 millimetres in diameter. It had 2 drillings, perpendicular holes going through it at right angles to each other. It was worked out that in all probability, if it did contain a poison, it could contain probably just 0.5 of a milligram of substance.
The question then was what sort of poison. All the toxicological analyses were negative. They got onto it pretty quickly because they had experience from work which had been done mainly behind the Iron Curtain of ricin, which is the toxic component of the castor oil plant.
Ricin is about 10 times more toxic than the nerve agent GB. It's about something like 160 times more toxic than hydrogen cyanide. And I think it's around about 2,500 times more toxic than arsenic.
Basically, it killed him by stopping his heart. by impairing the conduction system of the heart. And then you get cardiac arrest. As a result of that, the blood flow to his brain ceased, and that he would be clinically dead.
Oh, I think I've been cured by it, definitely. You know, it didn't cure me immediately, but it made the other treatments that hadn't worked before, it made them work so much better that it seemed to knock it out, and I've been clear now for nearly 4 years.
There's a less sinister side to ricin. Used to target and poison particular kinds of cancer cells, it's a very different magic bullet to the pellet used to kill Georgy Markov.
Any drug that you take is a poison. Any drug that you can buy in any chemist is a poison. It's only a matter of degree of poison. And some, obviously for magic bullets, the tendency is to go for extremely poisonous substances.
The model itself doesn't tell us why it's toxic, but it gives us a clue as to how it exerts its toxic effect. In the model, the ricin is composed of 2 parts. In green was the B chain, and in blue was the A chain. The B chain is the part that helps to bind ricin to cells and get it inside. The A chain is the part which then exerts a toxic effect on those cells.
Now, if you split those two apart and take the one that binds to the cell surface and throw it away, the other bit in itself isn't poisonous. Then it doesn't take a lot of ingenuity to think, all right, I will then attach the poisonous part with the thing that directs it to a particular cell. And when I take that particular thing, which is an antibody in this case, I then have a selective method of delivering the poison to a particular type of cell. It also had to have the properties whereby the poison that was attached to it was attached strongly enough while outside the cell to stay together, and then weakly enough that once it got inside the cell, they would fall apart and the poison could go about its poisonous activity. What we were not expecting were the side effects that occurred with it. A large number of patients found after a few days of having treatment with immunotoxin, they started to put on weight, they started to retain fluid, and their blood vessels became leaky.
Once the patient has lost between 1/2 and 2/3 of their blood volume quickly, the heart can no longer effectively get enough blood back to it to provide for the needs of the circulation. So each individual cell is getting less blood, and in a very short time, the heart won't even contract anymore because there isn't enough blood getting into the chambers for it to squeeze on, and the heart may stop dead. The heart begins to beat ineffectually. It doesn't know what to do. There's electricity coming from everywhere, and it can't pump enough blood, and so not enough circulation gets to the brain, and the person will die, sometimes within seconds.
Well, I've always liked happy endings, with all the loose ends tied up. Well, I thought that's what death did, and it closed everything down once and for all. But chemically speaking, it seems it's not quite that simple.
I think it's a taboo. I think the chemistry of human decomposition is a taboo. Not very many scientists work on it. It's quite hard to find a scientist that's interested in it. It's not clean and tidy, it's not pleasant. But we all come to it.
When the crypt of a church in Spitalfields in London had to be cleared to allow building work to start, a team of archaeologists, historians and scientists began working together to sift the evidence. What could the contents of the crypt tell them about how the people had lived and died? A key to the research was establishing when changes had occurred during life or as a result of chemical changes after death.
I think you've got to understand the processes that are involved. It isn't just a random soaking up of elements that are sitting there queuing up to come into the bone. They've got to be there in the first place.
like the high lead in the lead coffins and perhaps other elements coming out of the wooden coffins. But of course they're overlaying what was there in the diet. And I've got to try and tease one apart from the other. This is a child's face here, turned bright green. And we find from the records that they'd put copper pennies across his eyes after his death in order to keep the eyes closed. And as the body has decayed, as it's become more acid, the copper has been mobilized and has been taken up by the bone, gone right through inside the bone, and you get this bright, bright green skull. This looks like a perfectly normal skeleton of a child from the time, and we had no indication that there was anything peculiar about it until we started doing our analyses and x-raying it. And then we saw that there was some very remarkable differences. One day the radiographer came to me, really quite perturbed. I think the machine's gone wrong. And she showed me the x-ray. And here you see the picture she'd taken. And the lower part of the skeleton, she hadn't been able to get the x-rays to penetrate. They were completely white. We realized we had to take this further to try and identify what was causing the very opaque part of the bones. And by using the electron microprobe, you can actually map the different elements in different parts of the skeleton.
Right, well, this must be the edge of the bone along here. So this is just the sample stage at the top, and this is the surface of the top of the bone down below. So we can move a little bit further into the bone. It's imaging quite nicely. So we go right in the middle
so you
there,
choose
on
a
a
bright
nice
bit.
bright area. Okay. And then if we start the X-ray spectrum inquiry.
So the lead is right over the top.
The lead is enormous. The main component at the surface of this bone is the lead.
It was only when we realized that it was all the lower bones that were much more opaque than the upper bones, and that this could have been because the child had been lying in a coffin that was slightly tilted, and the lower part was in the coffin liquor, which is the liquid that forms in the coffin as the body decays, and would have been a means of transporting the lead from the lead of the coffin into the bones of the individual.
There is nothing supernatural about death. There is nothing that suddenly comes from elsewhere that is different from the way our bodies ordinarily function. Death occurs because we lose the possibility of oxygenating our cells. A particular kind of chemical reaction is stopped. It is a very natural phenomenon.